I welcome all the participants and our today's speaker, uh, Professor Naresh Kumar sir. As you all know that this is free science webinar series, especially in bioscience system, we are organizing regularly this without taking any fee, and we also request a speaker to means without charging any fee. Speakers are also delivering lecture. So today's speaker is my you can say mentor also. He has served 35 years at Department of Biochemistry, MS University, Baroda, in which he has also served as HOD several terms, and he has also served as Director Institute of Science in Nirma University, and he did his PhD from TIFR and several PhD. He has guided several PhD students and several research papers, review papers he has published. I have also used his lab during my stay at MS University, Baroda, to complete my fast track young scientist project. I have also served 2 years there, so I used his lab. So, with this brief introduction, I welcome sir in this science dialogue series. I heartily welcome you, and we are grateful that you are ready to deliver lecture in this free science webinar series without any branding, because general scientists are trying to deliver lecture on good brands, IIT, JNU and all these things. So there is no brand still. So people are ready to deliver. So I'm grateful to all the speakers, all the resource person who are willing to disseminate his or her knowledge without any using the branded platform. So thank you, sir. Now
Yeah.
platform is yours, sir. वो
Yeah.
विंडो किया इसीलिए इंटायर स्क्रीन में नहीं प्रॉब्लम आता सर.
Okay. Yeah, did the window. That's the mistake. Yeah.
Yes.
Okay. Uh,
Yes, sir.
I'm going to talk today on the preventive and reversal strategies for managing metabolic syndrome. Main reason is that our country and globally the uh, disease pattern has changed. Previously, it was more of infectious diseases, but now it has become what is called the non-communicable diseases. In that, diabetes, cardiovascular diseases, chronic respiratory diseases, cancer, and all that thing come. All of us are very much heard about cancer. You can see the latest data from our country. These about 15 lakh cases are there with the cancer. But if you look at the other diseases, the asthma and the chronic respiratory disease, that is 1.8 crores. And diabetes is 7.7 crores. And if you look at the cardiovascular diseases, 30% of our adults have the disease, and about 30 lakh deaths are there in 2024. So that means these diseases are becoming much more common in the present society in India. So we need to really address this as a serious problem. And people have also seen the latest report in Lancet, which also predicts how these things are going to grow. So the main problem with all these diseases are more complicated. I'll tell you something like this. These are what is the basis for this metabolic disease disorders? All these things are categorized as metabolic disorders. The main problem with this, the time scale, and they have some sort of a common features. If you look at the type 2 diabetes, this is the pattern you find, and the zero is the time scale where you are looking the diagnosis is made that one has diabetes, and you can see that the DC 10 years before the actual disease started, and by the time the disease is diagnosed, about 40% of our beta cell functions are abnormal. And then the disease progresses slowly over the next 15, 20 years, and this is going to become more complicated. So the problem is that by the time we diagnose the disease, already the disease progressed quite significantly. And this is an example. And the same sort of issue is there with other diseases, which is called the disease second metabolic disease is called the fatty liver, and this also has a similar pattern. It starts slowly over the period, and again it goes on the almost 25 years on so on and so forth. And here there are a lot of grades. Most of the time, by the time we diagnose it, it is already across the fibrosis stage 2 or something like that. Then third, what you said, the cardiovascular problem. This is mainly because of uh, the plaque formation in the blood vessels, and this again has a time scale of, we can say, around 40 years, and this is the most common thing present in the society today. So all these metabolic disorders have a common pattern. That is, by the time the disease is diagnosed, already disease has progressed to uh, quite large time scale. So, we- although we have some treatment and understanding of the diseases after the metabolic disease has been diagnosed, till now we don't have any cure. That is a one problem. Second, the main idea, the current perspective is that the prevention and is much more important than tracking with the diseases, because now we already, the prognosis is that the population is going to increase in all these diseases in large numbers. So we need to have some sort of a prevention. For prevention, we need to have very early stages of diagnosis. So current stage of diagnosis are not good enough.
सर वहाँ
So,
एक है वो प्रेजेंटेशन हाइड कर दीजिए, हाइड पर क्लिक कर दीजिए, हट जाए, वो लाइन बीच में आ रहा है.
oh, okay.
जस्ट हाइड, हाइड पर क्लिक करिएगा.
Okay.
यस
Okay.
सर.
Yeah. So the what clinicians started doing is they started looking for some sort of a uh, uh, um, physiological abnormalities, which can give some sort of an idea about the disease which is going to come at the early stages. So the most of the diseases are diagnosed at the 5 years or 10 years of the onset of the disease. Can we do something before that? So then they started looking the some sort of a idea. This is called the syndrome. Syndrome is a collection of symptoms that may not have any disease association or cause, but these physiological abnormalities are there. So that idea they have taken, and this called it as a metabolic syndrome. So the metabolic syndrome is a set of features, and this will be progressing into a specific metabolic disease, depends upon an individual. There is no clear idea about which disease one is going to get in. That may be based on the genetic predisposition as well as lifestyle and many other parameters. But this status is going to indicate in the early stage, much early stage, the physiology is in it slightly abnormal. This is what is called a metabolic syndrome. Now I am going to concentrate on the features of the metabolic syndrome and concentrate half of my talk on the how this metabolic syndrome is generated, and then we'll try to see some of the patterns which are emerging to tackle this metabolic syndrome. So general features are in terms of obesity, that is basically the belly fat, how much is accumulated, and this seems to be some way correlated with the lipid profile, that is the high triglycerides and the low level of HDL. HDL is a good cholesterol, high density lipoproteins. And again, there are two more features are there, which is called the elevated blood pressure and high blood sugar. These are the features which come at the these features are present either all of them or three are at least three parameters are there. One can call it as a metabolic syndrome. This is a status we will progress into slowly into a specific metabolic disease. So the the obesity features, people have tried to utilize many parameters to quantify this and bring some sort of a correlation with the disease. In this, what the complication has come, initially people used to use body mass index. That you have been hearing about, there's not a very good indication, because in our adipose, there are two kinds of adipose tissue. One, it is called the subcutaneous adipose tissue, that is in the outside, uh, that is in the hip, the belly, and legs, all these things with the fat which is present, that's called the subcutaneous thing. And there are in our body, their organs are present, the visceral organs. Around that, there is a separate type of adipose tissue is there. This is called visceral adipose. And what our data says is that visceral adipose tissue is the major culprit. The people, when they're healthy, they do not have much amount of visceral adipose tissue. As they become more diseases, in most of these metabolic diseases, people have shown visceral adipose tissue is more. So you can make some sort of a correlation that amount of visceral adipose tissue is present, that is going to be connected with the disease progression. So now people are trying to see, you can make some sort of a physical parameter to estimate this visceral adipose amount, how much is there. So these are the various parameters are there: body shape index, body adiposity index, various parameters. And what it appears in the males, the uh, waist waist circumference, followed by the height parameter ratio. That is one thing. This is second parameter is called waist circumference, the hip ratio. These two seem to be better than the BMI, and there is a uh, body uh, um, roundness index, the overall thing that's called a roundness index. These three are the good parameters, seem to be correlating with the disease progression. Whereas BMI, which is commonly used, that is not a good indication. This is the latest paper which has come. So we need to be some sort of a idea get we can have the our health status based on these parameters. Then what happens is, if you go into the physiological parameters, what people have seen is that initially the much earlier stage of this disease progression in the body, one has the insulin levels are high. Previously, people used to think insulin levels is only correlated with the diabetes, but now the most of the metabolic diseases much before you have a higher amount of insulin. So this is what is called the hyperinsulinemia. This is the so in the adipose, the obesity, initial stages when the visceral adipose tissue is less, we have seen the parameters before. If they are good, then the you don't have a problem. Then it changes, continues to where the visceral adipose tissue comes. Then the insulin levels also blood increases. The and along with this, so when you generate more amount of visceral adipose tissue, then two more features come. What is called the hyperleptinemia. The leptin is the peptide hormone which brings you when you eat the food, it gives you satiation. That is generally what happens. The fasting condition, this is low. After when you eat, the leptin levels increase, and it stops you eating food. Whereas in this case, the leptin levels, basal level also increases. So this is called a hyperleptinemia. Similar to that, there is also the ferritin level. Ferritin is a thing, it binds to iron. Iron deposition takes place inside the cell by ferritin. So if you have high amount of ferritin in a particular tissue, is the ferritin in the inside the cells is stored as a ferrous. Ferrous form is very, very susceptible to oxidation. So it generates a lot of reactive oxygens. So this is another problem. So these are the three recently people have realized hyperinsulinemia, hyperleptinemia, hyperferritinemia happens in the case, and this is the onset of metabolic syndrome. And once you have a basal levels of all these things are high, the tissues, when they are supposed to respond normally, basal levels are low. When the physiological condition, when the food intake or whatever the stimulus condition, they're supposed to increase and tissue is supposed to respond. That type of thing does not happen. So this is what is called the resistance. So once you have all this hyperleptinemia, hyperferritinemia, hyperinsulinemia, you also start getting insulin resistance, leptin resistance, and hepcidin resistance. Hepcidin is correlated with the ferritin secretion. So that is how the metabolic syndrome is progressing. After this is what is the onset of metabolic syndrome. Then it goes to the specific metabolic disease, depends upon the individual. Okay. And all these features, why do they happen? When do they happen? These are uh, determined by basically lifestyle. Lifestyle can be sleep, stress, exercise, and aging. uh, all of us know all these are very, very important features which contribute naturally for everything. But the sleep and stress disorders have increased. The sleep uh, pattern is changed with the society now, and stress also increased by the normal lifestyle, and exercise has gone down, and aging anyway is there. So all these parameters are contributing to more. So in this aging, the important aspect is that our lifespan is increasing. So the lifespan is increasing, we are also the metabolic disease are also progressing. So these are the general features. These are controlled or influenced by the many other parameters. What is called the diet. Diet is a crucial thing which controls this uh, physiological parameters. And in that, in our body, in the intestines, we also have large number of microbiota. This is called a gut microbiota. Diet and gut microbiota, there is a strong associations are there. So good diet is going to help good microbiota to survive and facilitate good health. If there is not a good diet, that is a too much of sugars, too much of fat, too much of carbohydrate, too much of alcohol, all these things are going to contribute to the dysfunction of the microbe, and everything is there. And the third case is what's called the inflammation and ROS. The cells are damaged. The inflammation is a part of immune responses. Immune responses generally are good for our body to take care of the tissue homeostasis to ensure cells are in a good shape, and the total tissue is in a good way functioning. So the inflammation tells you how much amount of damage is there. The immune cells will take care of the thing and help in the tissue functioning. So, and reactive oxygen species is depends upon the metabolism. So, any sort of metabolic dysfunctions are uh, uh, mediated in conjunction with the inflammation. The lot of cellular process, what we know as autophagy and apoptosis, all these things are taking place inside the normal tissue to take care of the proper maintenance of the tissue. But if this thing, there are two types of damages are present. One, what we know is the inflammation nowadays when they call the acute or damage response. That is when you have some sort of a tissue damage directly or pathogen infection. All these things are acute response. That in that is the levels of inflammatory cytokines and all the immune response are in a very high level, which help in the tissue protection where the wound repair is there, and the immune system takes care that whatever is external pathogen is removed. In addition to that, there is something called the chronic inflammation. This happens at a very low level. something like 100 times less than the acute response. And this is generally involved in the tissue maintenance. When there is no external pathogens and external tissue damage, the normal low level of inflammation is an indicator of the cells damage and repair and things going on. But if this type of inflammation continues for a very long time, that is again, I told you, caused by the various abnormalities of diet and microbiota relations or stress and all kinds of features. If it continues for a very long time, this is very important. This time scale we are talking in terms of something like 6 months or more. Low level of thing. We don't realize if you continuously keep doing the sleeplessness or having stress for 6 months or something like that, then there is going to become a chronic. That type of thing is going to set up whatever the abnormalities, what we talk about, the metabolic syndrome. Okay. So the and one of the main aspect of this happens, the our GA tract plays a very important role in initiation of this, because already I told you diet and microbiota have a major role in determining the metabolic syndrome initiation. This is associated with leaky gut and visceral adipose tissue. Now, what I'm going to show you is that our understanding has reached such a level that mostly the inflammatory intestinal damage is a thing which is responsible for visceral adipose tissue formation. So, this is the normal type of intestinal epithelium. This is the epithelium. This is the layer called mucus layer. And here you have large number of microorganisms. And these microorganisms are equivalent to number of cells in our body or even more than that. A large variety is there. And that population is dynamic and keeps changing, but all of them don't interact with the directly with our body cells and everything, is by protected by this mucus layer. In this, you have lot of peptides which are produced, which are bactericidal, so they don't allow any bacteria to come inside. And we also make these IgA antibodies, mucosal thing, which protects that. And these immune response are mediated by low level of TNF and pro-inflammatory cytokines are produced in low level. So this is responsible for making the IgE production and so on and so forth, the complete immune responses. An interesting aspect is these epithelial cells have very short time scale, only 3 to 5 days. And we're talking about that damage that has to be continuous, because these things are going off. Damage, the agent has to be continuous. Otherwise, the new layer has come, whatever the previous damage is already gone. So that is why the chronic inflammation is a very important feature of, because the epithelial cells have very short time scale. So, and we good, normally good bacteria, they produce a lot of things and they help the thing that is involved in the short chain fatty acids and bile acid metabolism. All of the things are helpful in the good way. But if you have some sort of a uh, bad bacteria, then they're going to ethanol or a lot of fructose. If you take, there are many agents which cause ROS generation inside the intestine. Those are the things which are going to create a problem in there. So you can see here that the emulsifiers, ethanol, and all the many other factors are going to influence the epithelial barrier. And some of the bacterial things, the prebiotics are some things which are necessary for the microbiome to survive. If they are not there, the bacteria start eating the mucin layer. So there are multiple reasons are there for why this epithelial barrier becomes a more permeable, and this permeability increases, then the microbial components, one of the major important problem of the lipopolysaccharide, which is involved in the immune activation. This caused them one of the major component of immune problem. So this is the status where the uh, um, damage chronic thing. We are not talking about again acute damage. We are talking about low level of ROS damage and permeability barrier changes. So these normally you see the intestinal things are very tight. Nothing will go. Even water cannot go between the cells. It will go only taken up by the cells in a coordinated fashion. But when this here epithelial barrier is broken, then all these things start coming inside and interacting with the cells and creating a problem. So the major problem is that we have the entire human intestine is about 320 square foot, very large thing, and and each region has different functions. Small intestine is mainly involved in the food digestion and absorption, whereas the colon is the thing which is involved in the iron absorption and water absorption. And the number of the environment is also different. Colon has a large number of microorganisms, where the small intestine has a small number. So we don't know actually, although we know that there is a intestinal barrier is a problem around this 320 square foot region, which region where it is happening, and is it depends upon the type of in different individuals, is it the same location which is gets affected or different thing, that also we don't know. But why it is important, I'll tell you later. the so this is the difference between what I was talking about is homeostasis. When the intestinal barrier is good, microbiota is good, you have short chain fatty acids, everything is nice, and you have what IL-20 and everything is anti-inflammatory, TGF beta, everything is good. When acute inflammation is a very strong pathogen, is there diarrhea type of thing is happening, there is the completely cells are broken here. This is the acute inflammation, but we are not talking about this. We are talking about chronic inflammation, slow, low level. It is the much less. The compared to these two, the chronic inflammation has something like 100-fold less inflammatory cytokines. Although inflammatory cytokines are same in acute and chronic, but their concentration is much different. So the response are different. So, how do we monitor this? I said around 6 months or more, you need the damage. The whatever we are taking here. This is a very good experiment. People have shown high fatty diet. People have gone to the animals and seen what happens. You can see this high fatty diet is 6 weeks are given. The in the case, what happens is the intestinal thing, the associated with it is a specific lymph system. This is called a mesenteric lymphatic system. So what happens is that all the lipids are taken through because these are not water soluble. They have to be transported through lymphatic system through what is called a chylomicrons. Big particles are made, and these are transported through lymphatic system. So lymphatic system is very important for high fat diet transport. So these you can see some of these things are there. The green layer. These are the case where the lymphatic vessels are here, and uh, if you keep taking the high fat diet longer time, then what happens is there is some sort of a problem. That is, normally the vessels from the intestine to the lymph node, whatever is the nearest lymph node, the lymphatic vessels are straight. But if the high fat diet is there, I already said, because of the high fat diet, the barrier is broken. Once the barrier is broken, LPS comes in. LPS interacts with the cells and creates a problem. So that makes the damage of the lymphatic vessels. And there is a damage and repair is taking place. What happens is, instead of straight one, they become convoluted long thing. That is called, you can see various places. This is called the instead of straight line lymphatic vessel becomes very much curved. Length increases. That is called tortuosity. Tortuosity is the length of the lymphatic vessel. From the linear one, how much is the ratio is there, that is going to tell you the amount of damage. So you can see that from 6 weeks, the damage is less. And by 15 weeks, the damage is increased, the branching and everything happens. The 15 and 32. So we are talking about very large time scale. And then what happens? These I told you the lymphatics is going to carry the fat, dietary fat in kilomicron. If the things are the lymphatics are damaged, the fat comes out of the lymphatic vessels and accumulates outside. When this fat is outside, then the immune cells come and form a cluster. This is called fat associated lymphatic clusters. This is the lymphatic vessel, and you can see that lymphatic clusters is there. And this again, you can see that these clusters basically present a low high fat diet is increased to by 15 weeks. Now they have increased, and by 32 weeks become very large. So to the damage of the lymphatics over the long time, the fatty acid, the chylomicron fatty acids are spilling out, and this is happening. So now this is the overview of what is happening about that. And once these things, LPS is there. LPS is the major problem. Why LPS accumulates more? That also the people have identified. So basically, when there is a epithelial barrier leakage, the mesenteric lymph node is supposed to be straight. This lymphatic damage thing, this becomes more and more convoluted, and in the process, lot of fat comes out and forms that the fat lymphoid clusters are formed. Once these things are going to now I'm going to show you here. I'll give you later more. This is the adipose tissue, what we call the visceral adipose. These things, what we are showing here, stomach and everything, these the intestines are here. From the intestine, we are talking about the fat accumulating here, and that is triggering more amount of problem. This is the adipose tissue here. So constant leakage from the chylomicrons, damage because of the epithelial barrier is broken for a long time, and that is the fat is coming out, and that fat is forming clusters, and that clusters is triggering the visceral adipose tissue formation. Once a visceral adipose tissue is formed more, then it is going to trigger what is called hyperthermia. I'll tell you how it happens. So this is what I was telling you. The which region of the intestines is important. You can see that where you have the colon, ileum, different places, slightly variation in the structures of villi is there, and you can see that there are the lymph nodes, mesenteric lymph node. There are many different lymph nodes are present, and the from the intestines, different regions go to the different mesenteric lymph node. You can see that how the cell, our intestine colon is going, the intestine is going, and here there's so many nodes are there. So the damage wherever it is present, and immediately that particular lymph node is going to be inflamed. So then what happens is when what we mean by inflamed is it makes more amount of immune cells to come together, so that node going to become bigger and bigger. Normally, the mesenteric lymph nodes are present in a very small size, about maybe millimeters in size. But as they inflame, they can become bigger and bigger. So if we are able to look at this by some sort of a MRI scanner, something like that, in the individuals, we can clearly find out which lymph node is getting inflamed. So although we said the epithelial layer is very dynamic, but damage which is happening in the particular region, this lymph node thing is going to be chronic thing. How much continuously damage is there, that lymph node is going to be a signature of what is happening. So this is the another way of trying to show you exactly what happens. We have this is all our intestines, the stomach, liver, and the duodenum here. So what is these things are, whenever there is a leakage here, the fat is coming out here. And between these organs, around that, all these organs are bound by a membrane. This is called the peritoneum. Entire thing is peritoneum. That peritoneum is very big. It is about 50% of our surface body area, and these are made up of separate cells called mesothelial cells, and these mesothelial cells are mesenchymal stem cells, and adipose tissue and adipocytes and the mesothelial cells are the same origin. So what happens is, if these cells get damaged, then there is a process called the transdifferentiation. They can undergo dedifferentiation and become more amount of adipose tissue. So what is being believed is that these are called the mesothelial cells. These mesothelial cells, because of constant inflammation which is exposed by LPS encountering all the things, those generate through these visceral organs. So this peritoneum is contributing to the visceral adipose tissue differentiation. These mesothelial cells are important, and sometime later you can see that these organs are very, very interesting. Uh, which mesentery we are talking about, the lymph nodes and everything are present in this mesentery. So suppose here the lymph node is present, here present, here present. This, although seeing the two-dimensional, three-dimensionally this is a very large area. Here this is distance is about 15 centimeters, but if you look this area, this total length, this comes about 15 feet. So it's, they saw something like a Japanese skirt, which is a, if you enter the, so the problems are having here. So if you are able to find out exactly where the node is getting affected, we can find out probably what is the reason and what is going on. That type of correlation still is not there. Okay. So the basically the first the intestinal problem, then it's a mesenchymal mesentery lymph system inflammation. Now we what I am saying is the peritoneum inflammation. This peritoneum inflammation is important for the transdifferentiation of mesothelial cells to form the visceral adipose tissue. This is the thing which is after this you can see that this is the thing which is the yellow color is the visceral adipose tissue. The pink color is the subcutaneous thing. So in the normal case, lean individuals, you can see that visceral adipose tissue is very less. The you have this is a pink one here. Over the 6 months or 1 year or something like that, this visceral adipose tissue keep increasing, and this is having the lot of inflammatory properties. This is going to contribute to the major problem. This is what is the metabolic syndrome. Once this visceral adipose tissue is set, then because all these organs I showed you, you can see that here, pancreas is also here. So pancreas and liver, they get affected, and that causes your hyperinsulinemia. So, interesting thing is the pancreas, when the insulin is produced, the it goes directly to the liver, and the liver, out of the whatever insulin can, 80% gets degraded, and only 20% goes into the body circulation for muscle and adipose tissue. But now the insulin blood levels are depends upon this 20%. But the higher insulin level can be found because either two reasons. One, the beta cells itself, they are producing more amount of insulin. Second, the amount of the removal of the insulin is called insulin clearance in the liver, that can be also decreased. So the inflammation coming from the portal blood, liver is getting affected, and visceral adipose tissue, everything is causing the pancreas. Net result of these two is that you're going to have more amount of insulin. Normally what happens, the pancreas produces only after food stimulus, glucose and all those amino acids, all those things that trigger this insulin secretion. But because of all these problems, more amount of insulin is present, is not getting eliminated. So that there are the features of the hyperinsulinemia. First, high amount of insulin, and the normally insulin is stored as a proinsulin, and then during the secretion gets converted into insulin. But that process is also becomes a problem. So you have more amount of proinsulin, and C peptide is supposed to this also increases. So there are so many problems are there with the hyper insulin secretion. So this all gives a problem for the amylin is also secreted along with the insulin. If you have high amount of amylin, it forms aggregates. And the normally amylin, which is there, it goes to the brain and controls the food intake. But if amylin forms aggregate, we are losing the food intake control. And additionally, because the amylin aggregate, they also cause inflammation. In this inflammation, what happens in the tissue? There are macrophages. Most of the macrophages are present for the tissue repair mechanism. Now, instead of that, there are the M2 ones. This transits to M1, so that becomes a more inflammation. So this is what is happening. Net amount of this hyperinsulinemia is lipolysis gives to obesity, uh, more amount of hunger generation, lethargy, all these things happen. So this is the hyperinsulinemic status, and this can give rise to the endothelial dysfunction is associated with the cardiovascular problem, the hepatic problem, the skeletal muscle problem is due to more amount of diabetes. If it is the liver problem, it's going to be fatty liver. So many diseases are going to come because of these consequences. Uh, Okay. So once you have the all this happen, now actually metabolic syndrome has taken place. Now where which individual what disease it progresses, we are not very clear. But this metabolic syndrome can give the precursor for all these diseases. It can be PCOS, diabetes, all other diseases. So this is the first state which is coming there. Okay. Now I'll look at the second half thing where we really going to how to prevent this. We the normal thing what we know is the diet, exercise. These are the things that which are known. For the diet, you have three. One thing you can do calorie restriction, eat something less. Then is the nature of the food and time of food. There are three parameters here. Exercise, there are two types of exercises, endurance and resistance. Um, then these are the strategies. Then third one, you can have some sort without doing this, can we think of alternative strategies? So diet management, people have done the what called the diet restriction. There, one is called the intermittent fasting. In this, the two models people have done. One is the alternate day fasting, or within a week, 2 days you don't eat, 5 days. But and the third thing is called a time restricted eating. In this, you have a window of eating. You have 4 to 12 hours is the eating time. You can see the pattern here. This is the alternative day. One day eat, one the day fasting, like that. This is the 2 days in a week. You adjust Sunday and Wednesday, something like that. That is another pattern. Or either two different days or two consecutive days. Uh, this is called a time restricted pattern. In this, they have various things where you eat. Eating time is here 4 hours, 6 hours, 8 hours, 10 hours, and this period, fasting period, you don't eat anything. So these are the things people have been doing, and in these things, there are a lot of adverse effects people have found. So better thing is to have this type of pattern where you eat for some time and then fast for longer time. Whatever is convenient for people can follow. Better things are in this compared to fasting pattern. And in this, people have shown all kinds of effect advantages are there. you find the body weight, all the parameters are going to be good. Whatever you are doing, one can follow that. Uh, that is one way of it. So this is what is called a chrononutrition. It benefits overall health of the individual. Then something has come because the carbohydrate, insulin, already hyperinsulinemia is there. If you use more amount of carbohydrate, it is going to trigger more amount of insulin. So people thought instead of this, we go for more amount of keto diet. The uh, uh, different types of keto diets are there, where it is 4 is to 1. The fat is very high, carbohydrate is only one ratio. Here, the another thing is that is there large amount of energy comes from fat instead of um, carbohydrate. So the people have tried different type of thing. So the basic idea here is that you are not triggering um, insulin secretion, whatever the things are involved in that. So people have shown the ketogenic defect. Uh, Also, because of the sugar is not the inflammation will decrease. Some beneficial effects people have found, but the problem is the diet has to be controlled quite seriously. The glucose levels have to be monitored very carefully. So mechanistically, what people have shown is that in the ketone bodies are increased when the fatty acids are get oxidized. You have acetyl acetone. This is a ketone body. This gets converted into what's called the 3-hydroxy butyric acid. This butyric acid is secreted into blood. These are the ketone bodies present in the blood: the acetyl acetone, acetone, and beta hydroxybutyric acid. These things go to the muscle. Muscle can utilize, interestingly, muscle and brain can utilize these ketone bodies and generate energy. So you don't need to give glucose. By using these ketone bodies, they can. So this is what is the basis of the keto diet thing. So what uh, they wanted to know how this ketone bodies are helping the obesity, the reversal, insulin resistance, and all that thing. So the what they did is they gave a compound butanediol in the diet, and this, when it is taken by the animal in the liver, it gets converted to 3-hydroxybutyrate. So you don't need to have a keto diet. This particular compound is given, it goes to 3-hydroxybutyrate, and 3-hydroxybutyrate is sufficient to initiate good responses. So what happens is there is a transcriptional activator called the PPAR GABA, peroxisome proliferator activator receptor gamma. This is involved in the expression of adiponectin, leptin, and GLUT4 in the adipose tissue, and these are going to make, if they are produced, they're producing insulin sensitivity. So the normally what happens, the glucose, insulin is supposed to trigger on this and make the thing, but in the diabetes and the metabolic syndrome, the adipose tissue is going to become insulin resistant. These are not triggered on. That's how the thing sets in. So if you give 3-hydroxybutyrate, there is a signal transduction operate, and this PPR gamma is gets activated, and you produce this. This is how insulin sensitivity is triggered on the adipose tissue. Once this insulin, adiponectin, all these things are produced, then slowly the reversal happens. That is the mechanism here. This is different from the PPR analogs before, because here PPR gamma gets inactivated by serine phosphorylation normally. So even if you take analogs of the PPR gamma, they're not going to help. In this particular case, the serine phosphorylation is prevented by the 3-hydroxybutyrate, and this is going to help. So this is the mechanism by itself. Then people have talked about exercise, because exercise utilizing glucose, the muscle metabolism is completely independent of insulin. So you do this exercise and it's going to help. And people have shown that all kinds of exercise is going to be helpful. But uh, now, although one can do fasting and exercise and everything, uh, it may not be really practically possible. So, is it possible to do something mimicking things? In the case keto diet, without using, now we have seen that the 1,3-butane-diol is sufficient to do mimic the keto diet. Similarly, can we have something on exercise mimicking? This is the thing what I already told you. We don't need to do fasting or a ketogenic diet. You taking the butane-diol, we can mimic that. Second, now what happens is when you do exercise, lactic acid is produced more, and our body, what it does, the lactate interact with the phenylalanine. It forms a compound called lactyl phenyl. This is done by this particular enzyme. This is what is involved in the helping the muscle metabolism, everything more. It decreases the appetite, food intake, everything. So this compound is going to act on the brain. This is the one of the mechanisms of the exercise doing the benefit to our body. So what people have done is they have given a compound. This is called lac. This is a lactate plus butyric acid uh, compound analog. So this, when it is taken up inside, this get hydrolyzed, you get lactate and beta hydroxybutyrate. And this is going to, so the one compound is mimicking the exercise as well as the ketogenic diet. So this is the very, very nice compound. You are mimicking both the effects of diet as well as exercise using this particular thing. So you can see that this is the latest paper, and they have shown the ways how this is helping the uh, metabolism. So free fatty acids, everything has gone down. You can see that how the how the blood levels are increased by giving this compound. Uh, then there is something very interesting people have done. They found in the exercise the mimicking by there is new receptors are involved produced. These are called estrogen related receptors. When the exercise is produced, and these things are involved in the fatty acid oxidation. This is a transcription factor. These fractions in the muscle, there are two kinds of fiber, type 2 and type 1. Type 1 are the things which are fatty acid oxidation thing. So as you do more amount of exercise, the muscle type from type 2 to type 1 it changes. So these are the things which are produced. So now people are found in a analog of this compound. Interestingly, the none of the estrogen related compounds can bind to this and activate this. People don't know what is the natural ligand which can activate this receptor. So artificially they made, because using all these docking studies and everything, they made a compound, and this can bind to this and initiate the energy metabolism. So this is another very interesting. This is completely independent of what I told you, lactic acid and keto diet. This is a completely different strategy where the you can mimic the exercise by using this agonist. One more interesting thing has come. This is called abscisic acid. We know abscisic acid in plants, which is involved in the abiotic stress responses. Interestingly, people found abscisic acid receptors are present in the mammalian tissues, and these things are act by the binding to, again, this one way. This is a protein which binds to this, and this protein activates the estrogen related receptors and so on, increases the metabolic oxidation. So now you can have more amount of abscisic. So what they have done, instead of abscisic acid, created a artificial compound which is called the gamma oryzanol, and this is going to initiate that. So now we have 3-4 different strategies which is activating the energy. This is using another pathway, it's called the PGC1 alpha, which are in the mitochondrial metabolism and so on. So now the using this strategy, whatever is going on, the entire mitochondrial metabolism, people are thinking of generating small molecules which can mimic this exercise responses. Exercise responses, we said different, so many different things are present because we said the two kinds of exercises are there. So the different type of muscle responses are present. So we can create, mimic each kind of the exercise responses, and we can see which type is more suitable for an individual, whether the resistant type of exercise or aerobic or anaerobic exercise, which is favorable, that type of thing we can design. So this is what is going on now. If from mimicking things, reversal process, we can also do other things by using the hyperinsulinemia reversal. People have used mesenteric lymph targeted COX-2 inhibitors. We said that's what. And then you can use a lot of nutritional supplements. These are antioxidants, anti-inflammatory compounds, and of course you can have good probiotics with having antioxidant and inflammatory compounds. And interestingly, people have made vaccines against these amyloid plaques. Amyloid plaque, I told you, it's going to cause the islet inflammation and the problem that can be cured by this vaccine. So so, and we can monitoring of the thing already. Biochemical parameters is we can use insulin levels, proinsulin levels, C-peptide, amylin levels, so many other things which are coming. And we, if we use this, the location and extent of mesenteric lymphatic damage, we can probably find out uh, by using CT scan exactly what is the location per individual, and we can find this some sort of a causal relation with the diet or sleep or stress or what it is, and we can figure out what is going on. And then the we said most of the things are connected with the adipose tissue, metabolic syndrome setup. This recent paper says the VAT is main problem for men, and subcutaneous adipose tissue is for women. And interestingly, the adipose tissue is very susceptible for low temperatures. If you decrease the temperature, the adipose tissue, uh, these adipocytes degrade and you can remove the fat. So that type of thing is there for some sort of a cosmetic treatment is there for subcutaneous thing is going on. But these things are now, there are also some techniques which are coming to mimic the human fat. So there are the different strategies are taking place. So, what is the future? The uh, the main challenge is to understand the role of this peritoneum and mesentery in exact mechanism of visceral adipose tissue formation, and then developing prevention and reversal strategies for the peritoneum and mesentery, and optimizing this cryolipolysis is going to be very good. That is a challenge. And then in the good strategies for delivery of this antioxidant and anti-inflammatory. This is a chronic thing. They have to be there. We have now, but they are not a chronic. They are acute type of thing. Now we have to have an effective system for chronic thing. So this is the thing. So we are in a just nice phase of understanding the metabolic syndrome and some idea about how to address this problem. Thank you.
बहुत अच्छा है सर. पूरा मेटाबॉलिक पाथवे और मेटाबॉलिक सिंड्रोम का समरी एक हो गया. Those who are working उसके लिए ग्लिम्स मिल गए. Now platform is open for question answer. Yes sir,
Now
now you
stop
can stop
presenting.
sir. Yes sir, you can stop. Yes.
Okay.
Now anyone can ask question. I can see there are so many faculties here. शिखा जी, शिखा श्रीवास्तव, Ravi sir has also joined. है ना?
Yeah.
Anyone have any question related to metabolic disorders?
जी सर.
आप भी ज्वाइन कर लें, आप तो प्लांट पर कर रहे हैं.
सर एक्चुअली ये मेरा एरिया ऑफ इंटरेस्ट है. मतलब
Okay.
वैसे मैं बॉटनिस्ट हूँ, लेकिन ये क्योंकि आजकल इतना ज़्यादा प्रॉब्लम है नॉन कम्युनिकेबल डिजीजेस का कि स्टूडेंट्स को काउंसिल करने में हेल्प होता है थोड़ा सा. तो ये मैं पढ़ती रहती हूँ एडिशनल इन्फॉर्मेशन इसमें रिलेटेड मेटाबॉलिक सिंड्रोम से रिलेटेड जो डिसऑर्डर्स हैं. तो उसके बेसिस पे थोड़ी हेल्प मिलती है. सर का प्रेजेंटेशन बहुत अच्छा था एंड इट वाज वेरी इन्फॉर्मेटिव. बहुत सारा अपडेशन हुआ नॉलेज में.
Sikhaji is teaching in Delhi University, sir.
Oh, okay. Interesting. Yeah.
भास्कराचार्य कॉलेज में पढ़ाती हैं मैडम.
Oh, okay. Good, good, good.
Any? Yes, Ravi ji, your mic is also on.
Yes. I have a lot of comments anyway, but
बोलिए सर, बोलिए. This
So-
is interactive session. This is not one way. हमने इसको साइंस डायलॉग सीरीज को इंटरेक्टिव सेशन रखा है ना, so that researcher can interact, ask question, cross questioning, debate, discussion, so that knowledge will है ना improve and
Right.
percolate.
Right. So
Anyone have any question?
anyway, I was thinking that in terms of just not so much on the reversal or even fixing, but just to control hyperinsulinemia. Right now, people are trying randomly different combination of therapeutics, but I think if we measure the metabolites properly and choose what mechanisms we want to address, so and see where different damages are there. For example, should we be going with glitazones for addressing PPAR gamma, or metformin, or sulfonylureas, or combination, and all that. And now one area, this SGLT2 inhibitors, where you are simply removing sugar. Now, is really reducing hyperinsulinemia, reversing any of the metabolic syndrome symptoms, is one of the key questions that we have to focus, because right now all the therapy is only on hyperinsulinemia, hyperglycemia, so, and controlling that. And but there may be some mechanisms where by reducing blood sugar also is somehow reversing, or is it not? I mean, this is one of the key questions we have to answer, so that we don't indiscriminately keep on reducing blood sugar. So that's just one thought that I have.
So I request Sir Ravi sir, please introduce, and then sir will reply, so that participants will also come to know who is asking.
Oh,
Ravi?
okay.
Yes.
Sir, yeah, I am fortunate to be studying and working with Professor Naresh, and my name is Dr. Ravi Muppirala.
So you both have done PhD from TIFR, है ना?
Yes.
So yes. Now it's good you are asking question and sir is replying, so other will है ना, वो knowledge जो है ना वो जाएगा, उसका जो झरेगा knowledge तो सबको मिलेगा थोड़ा सा. Sir.
Yeah. This all these mimic exercise mimetics and keto diet mimetics are completely approaching the different level. They are doing on the adipose tissue and the metabolism, nothing connected with the glucose level. So that's why it is a very interesting strategy which is emerging.
Right. But I think, as you already said, we probably have to choose which approach at what time. Because if there is already ER stress and beta cell damage, what we can do, or something can be reversed. Now, fixing lipid metabolism, can it bring it back to some reversal? All these issues, because we have to fix, we have to try to also target what is already damaged. So, for people who are at a later stage of diabetes.
I think first strategy is to decrease the adipose tissue.
Yeah.
That's why I said that cryolipolysis has a great future. Then supplementarily, you have to protect the antioxidant, dietary things,
Yeah.
antioxidants,
No, that I
anti-inflammatory.
agree is a very critical
And
thing.
along with this, these exercise and diet mimetics, they all this. First, these are the strategy. First, you have to take care of the adipose tissue by cryolipolysis. Then you follow the antioxidant, anti-inflammatory things, and exercise and diet mimetics. I think that is a good strategy for reversal as well as prevention.
Okay, thank you.
Sanjay sir is also I'm able to. संजय झा जी, संजय जी हैं, दिख रहे हैं मुझे अभी, वही संजय जी हैं, संजय झा. Okay. Any other participants have any question? सर ट्रेन में संजय झा जी हैं, ये कहाँ हैं? No question. I have shared feedback link also in chat box. Those who have not joined any of our social sites, so you can join so that you will not miss next lecture. There is lecture on 24th also, 25th also, on 2nd May, 3rd May, 8th May. So if there is no more question, then from my side, sir, thank you very much for this insightful lecture. And there are so many, sometimes the students are hesitant, but थोड़ा तो प्राइमिंग होगा, then in future they will understand. क्योंकि पहली बार सब कुछ समझना बहुत सारे स्टूडेंट के लिए मुश्किल है. But some are research scholar also, some are faculties also. So in this way I am feeling successful कि चलो थोड़ा सा तो नॉलेज पर्कुलेशन हो रहा है, क्योंकि ऑफलाइन मोड में इतना टाइम भी नहीं मिलता है. क्योंकि आई हैव सीन कि 30 मिनट से ज्यादा किसी इवन की नोट स्पीकर को भी 30-40 मिनट से ज्यादा नहीं मिलता है. एंड विद इन दैट टाइम व्हेन दे स्टार्ट कॉन्फ्रेंस लेट, सो दे कट शॉर्ट दैट, सो दे डोंट अलाउ क्वेश्चन आंसर in most of the conference. So here people can utilize this interactive session, and I'm really grateful to all the speakers who are willing to deliver lecture, because it's a very daring task to deliver lecture online, because we are not, means the speakers are not seeing who are this side that side. ये बहुत साहस की बात है ऑनलाइन में लेक्चर लेना, क्योंकि क्लासरूम में लेक्चर लेना आसान है, ऑल आर फ्रॉम सेम ग्रुप है ना, और उसमें कुछ होगा. बट ऑनलाइन आई हैव सीन कि so many people are hesitant to take online. So I'm trying to this exercise is also good कि इसमें जो भीड़ है टीचर्स का, उसमें से those who are taking online means ये एक indirect establishment हो जा रहा है कि yes they are taking that topic very है ना bravely. And they are doing.
सर
जी
सॉरी टू
मैडम,
इंटरप्ट यू, मैं आप अलाउ करें तो मैं एक क्वेश्चन पूछूं सर
बोलिए
से.
बोलिए मैडम बोलिए. हाँ इंट्रोडक्शन तो हो चुका है. यस मैडम.
सर बीच में थोड़ी देर के लिए मेरा इंटरनेट कनेक्टिविटी, जैसे आपकी स्लाइड नहीं दिखी थी, तो मे बी आपने वो स्लाइड दिखाई हो, लेकिन मतलब मुझे थोड़ा सा इस पे इंटरैक्ट करना है आपसे कि जो ये मेटाबॉलिक सिंड्रोम है, इसका डिसबायोसिस से कोई रिलेशन है क्या डायरेक्ट?
Dysbiosis is the major problem. So what I told you, lipopolysaccharide is the major problem. And lipopolysaccharide is the component, cell wall components of gram-negative bacteria. So when there is because of whatever the reasons, the gram-negative bacterial number increases, and there is a damage of the intestinal epithelium, this is going to accentuate it. So that is a direct connection is there with the microbiota and this. And secondly, the good bacteria also have to go down. Short chain fatty acids are good for the maintenance of the intestinal integrity. If they go down, that is also creates a problem. Then there are bacteria which are in mucin they digest the mucin if they don't have any other alternative carbon source. So that's the problem. So the prebiotics are the thing. If it is not there in the food, then the bacteria start eating the mucin. So these are the main reasons how uh, dysbiosis creates a problem, and that has to be not a acute. It has to be over 6 months or greater time. So that's the problem there, because the disease offset, all these things are taking place years of time scale. Reversal also is going to take that long time. Okay, we might be able to accelerate a little bit, but not too fast. You can't expect in 1 month everything to reverse. It's not possible.
So, sir, you answered my next question also. So, my next question was related to it only. That can it be reversed with the use of probiotics and all?
Yes. Yeah.
Uh, that can be a sustainable approach towards this reversal.
Yeah, but what is very interesting is I was really fascinated when the abscisic acid is controlling the energy metabolism of mammalian cells, and you have a protein which binds to it, and transcriptional level it is controlling. Amazing, amazing. I read more so much about abscisic acid in bacteria and plants, not in mammals.
So, sir, shall I start recommending my students to take more, add more prebiotics in their diet?
Of course.
So that
Yeah,
was
yeah.
a take of.
Yeah, that's sure. Sure. More amount of the one important thing is that people think that curd eating is good enough, cheese and curd, but that's not true. These probiotics have a special feature. Probiotics and prebiotic combination is very good.
Okay. Thank you so much, sir.
Any participants have any complementation, comment, suggestion regarding today's talk or this science dialogue series? You can share. Yes, Sarita Gupta, you have raised. Sarita Gupta, madam, hai ya? Hello.
हाँ सरिता गुप्ता मैडम है
Yes, madam. Welcome,
Hello.
madam.
I was happy to see you and very interesting talk. Very interesting. Even though I have been reading so much on metabolic syndrome, I have learned lot of things, mainly specifically with respect to gut,
Okay.
which we
Okay.
generally don't give too much. We talk about adipose, we talk about skeletal, we talk about brain and all that. So yes, it was very interesting. And
Thank you, madam, for joining. And
I
मैं भी बहुत लकी हूँ कि सारे बायोकेमिस्ट्री और जो हेड हैं सबका मैंने लेक्चर करवा लिया है गुजरात में। मैडम ने भी लिया स्टेम सेल पर में।
that I don't know whether Naresh has attended. शायद नहीं किया उसने.
सर नहीं शायद थे उसमें।
So, Sarita, what is interesting is that all these biomimetics, you don't need to do exercise. All the new things are coming. Very interesting.
But one thing I would tell you that even like the compound which we are working on, that is also showing lot of these things which you have talked about.
मैडम कैमरा ऑन नहीं हो रहा है आपका?
कैमरा तो ठीक है, मैं बात तो कर रही हूँ.
Maybe they are binding to the estrogen related receptor.
That I don't know, but the thing is, in one of our experiment, we have seen that the PCOS patients or PCOS condition is also getting improved. Okay, so
Yeah.
where we had this granulosa cell, insulin resistant granulosa cell. So what you said, insulin resistance, that is one of the major key for metabolic syndrome.
Yes. Yes.
It's a very important thing. And most of the time in PCOS patient, it is like they just give metformin. Okay.
Uh,
Now, many of the females actually do not have insulin resistance. That also we have seen.
okay.
That PCOD with insulin resistance and PCOD without insulin resistance.
Do they have that? See, one thing people have shown recently,
I'm
in the
trying to
adipose
say
tissue, you have lot of ferritin accumulation. and that creates
Yeah.
a problem.
See, the recommendation what we gave to the, because this has been discussed in gynecology conference.
Uh,
Okay, so we try to tell them that for PCOD patient, it is very important that you first check whether there is insulin resistance in the patient or not. And
okay.
according to that, the treatment should be given.
They have hyperinsulinemia?
Yeah, some have hyperinsulinemia.
Okay, okay.
But
That is a precondition, it appears.
tissue
Uh,
level differences, the cellular level differences, which could be different in different patients. So, what I learned from here, this thing, certainly these news about exercise and those mimetics, certainly very good, and one can think about it. But many of these small molecules are
yes.
already working on few of these places where what you have said.
Yeah, I'm telling you, abscisic acid and the estrogen-related receptor,
That
that especially natural ligand is not known.
estrogen, you said, that's why I talked about PCOD. So we
No,
have
no, but estrogen doesn't bind to that. That's a very interesting. This receptor is produced on the exercise, and it is controlling the energy metabolism. People don't know what is the natural receptor ligand.
for abscisic acid. Yeah, yeah, that is a new finding. That even
Not
I.
abscisic acid. Abscisic acid is the protein is there. I'm talking about estrogen related receptor. For that, all these estrogen analogs, they don't bind.
They will, because they it is known to be binding to estrogen receptor alpha or beta type.
No, no, this is a different related receptor, ERR alpha.
Okay,
These are
so
different from that.
maybe I will have to
They are
check.
only produced in the exercise.
Okay, 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 fine.
So that is why they made that artificially they synthesize the ligand, and they showed it is they are able to mimic that.
So Gopal, you have to stop, otherwise myself and Naresh will keep on talking.
I have, I have, I have one more. It's just my personal perspective. See, the thing is that we have been talking about physical activity and exercise for, should I say, at least 50 years for both cardiovascular issues, diabetes, obesity, and all that. But there is no compelling, conclusive evidence that whether it be all these things are interesting, exercise mimetics, exercise mechanisms, all that, but to what extent they are quantitatively effective, and can they be something that FDA can take it up seriously, can they be something that we can start mandating therapeutically. So we are not there yet. And there are several lobby groups, including people working in physical metabolic physiology, exercise physiology, and all that. Everybody has an access to grind to promote their things, especially in USA. But I mean, the thing is that I think we need to be a lot more focused on evidence and concrete evidence and quantitative evidence. So that is still, we are far from that, in my opinion.
Dr. Sanjay Jha has sent message, sir.
Oh,
He is in our Navsari Agriculture University, he is dean there, principal. Thank you, Dr. Naresh Kumar, sir, for engaging lecture. It was both insightful and thought-provoking. Your expertise made complex topic accessible and interesting. Thank you, Dr. Gopal ji, running such informative session. So thank you, Sanjay ji. I know you are traveling in this time, maybe through train. So this is good. एंड मेटाबॉलिक डिजॉर्डर पर मुझे लग रहा है कि एक पैनल रखना पड़ेगा डिस्कशन का और 4-5 घंटे का रखना पड़ेगा.
yeah, that's a very, very important. We need to, a lot of emphasis has to be done on this, because it's a very serious problem in
Yes.
India as well as world.
Yes, that is important, huh, Gopal ji.
यस, और कुछ डॉक्टर को भी मुझे लग रहा है जो मेटाबॉलिक उस पर कर रहे हैं उनको भी इंक्लूड करना पड़ेगा इस टाइप का
Yes.
वन डे, ऐसा अच्छा रहेगा.
Yes.
Thank you very much, sir, and all the participants, including Saita madam, Sanjay sir, Ravi sir, Sikha madam, and all the students. It's good. Multiple, many faculties are involving. They are interacting. It's good exercise. Thank you very much for making this more interactive. Good night. Thank you, sir.
Thank you, sir.
Okay.
Thank
Good
you, sir.
night. Good.